Good morning, and thank you for joining us. We welcome you to the ISS National Laboratory Rodent Research Reference Mission 2 webinar. Our goal today is to provide everyone in the research community who is interested in responding to this request for proposals some background on the mission concept, the experimental design, and give you an opportunity to post questions in the questions panel of your webinar window that you have with regard to the RFP and to the mission itself. This webinar is being recorded and will be available on the solicitation website. We will also make available questions and answers that we receive today or via email requesting information. Next slide. My name is Liz Warren. I am an Associate Program Scientist for the International Space Station ISS National Laboratory. I have the privilege today of introducing you to the ISS National Lab and discussing the rodent research mission concept. I will then hand it over to our collaborators in supporting this mission. We have today Dr. Louis Stodiak, the Center Director of Bioserve Space Technologies at the University of Colorado at Boulder, Dr. Gretchen Cusick, Associate Director for Scientific Services at Taconic Biosciences, Incorporated, and we'll close with Dr. Bill McClam, Operations Project Manager for the International Space Station ISS National Laboratory, who will address any questions as we see them coming in. Next slide. If you have questions that are not addressed today, please submit them via email to info@issnationallab.org. Answers will be posted on the solicitation website below around mid-April. Next slide. I wanted to take just a few minutes to introduce everyone to the ISS National Laboratory. Our mission itself is quite simple. We support research and technology development using the International Space Station to foster scientific discovery and technological innovation in space for the benefit of Earth. In managing the ISS National Laboratory, we work in close partnerships with NASA, a variety of commercial service providers and implementation partners, and all of those entities are intimately involved in the planning and execution of this rodent research reference mission. Next slide. We draw on the ISS National Lab science to improve the lives of people on Earth and provide value to the nation. So our mission in managing the ISS National Lab parallels that of NASA and of other federal agencies who fund research and technology development to address national goals. In focusing on the ISS National Lab, we also seek to create partnerships across disciplines and industries. Our rodent research reference missions are a very good example of these new partnerships that we are enabling in order to expand access to data and knowledge and biospecimens from the International Space Station National Lab. Next slide. So very quickly, why space? The particular model of interest here is access to continuous microgravity. We understand from previous research focused on astronaut crews, multiple animal models, and more recently on cell-based models, that exposure to microgravity has profound effects on the organism. And in some cases, an organism's response to microgravity mimic the effects of disease here on Earth, most noticeably effects on bone mineral density loss and muscle atrophy. But other organ systems are impacted in other ways, including immune dysfunction, including neurocognitive function. So what we are seeking to do with our rodent research reference missions is to build upon the accessible data that's available using animal models in this case rodents, for understanding ways in which microgravity serves as an accelerant to disease processes that are relevant to humans living on Earth. And then, in addition to microgravity, the ISS National Lab operates as an access point or exposure facility to the extreme environment of space. That's not of particular interest to most biological investigations, but has significant interest and advantages for those working in materials science and the technology sector. In addition, the International Space Station National Laboratory operates at a unique vantage point and offers opportunities for both remote Earth observation and observation of deep space. Next slide. So our purpose here today is to talk about our second rodent research reference mission, which will be launched, also known as Rodent Research 17, no earlier than July 8th of this year, 2019. In this particular request for proposals, we have focused on an application for spaceflight biospecimens. The concept here is based on creating an opportunity for investigators to obtain biospecimens from animals that have been exposed to spaceflight for the purposes of their own investigation. A variety of animal models have been flown to the ISS before, though we are seeking now to increase the validity of the data in the database and to validate the use of different strains by filling in the gaps, if you will, and to create a repository of specimens and data that will be publicly available to the research community. Rodent Research Reference Mission 2 is focused on studying the effects of microgravity and its impact on bone, muscle, and other organ systems in both young and old mice. These mice will be of the same strain, but two different cohorts. So the mice will experience all the same rigors of launch, living in space, and return to Earth and recovery. This is the only difference between the space-exposed mice and the ground controls, which will be housed in identical caging conditions. And the next speaker will have a lot more detail on that. We then hope that this data will enable the research community to understand more details about the onset and effects of aging and downstream disease processes. Next slide. And with that, I'd like to turn it over to Dr. Louis Stodiak from Bioserve Space Technologies, who's going to provide everyone with an overview of the Rodent Research Reference Mission, the specific concept of operations for this mission, and some information on previous rodent research missions flown to the ISS. Louis?
Thank you, Liz, and good morning, everybody. So as Liz mentioned, I'm going to start out by giving you an overview of the Rodent Research Reference Mission concept and approach and what we're trying to accomplish with it. So on this slide, basically, I'm just indicating and re-emphasizing what we're trying to do here. The goals, overall goals, are to develop, enable, and validate microgravity animal models, primarily for terrestrial disease applications, but obviously there are also space applications. We're trying to standardize an approach. We're trying to standardize data sets and how data is collected, tissues, how they're collected, the specific protocols, the timeline as much as possible, all with the goal of permitting and enabling cross-study comparisons to be made more precisely and more accurately. And we're trying to collect and preserve literally as many tissues as reasonably possible, both for dissemination now through this solicitation to people that are awarded these tissues, but also to collect and preserve and maintain for future applications as requests are made. Next slide, please. The general, for those that are not familiar with conducting animal rodent research on space station, this is the general flow that is followed. and in particular in regard to these reference missions, obviously with the partner that you'll hear from later at Taconic, we start in that lower middle box there. The animals are bred and born and raised initially at Taconic Biosciences. They are housed to maintain their specific pathogen-free status. That's a requirement from NASA. They are microchipped so that they can be individually ID'd. They are weighed, assessed, and then ultimately shipped to NASA Kennedy Space Center, going to the next box where they are received somewhere approximately 4 weeks ahead of the mission. They are acclimated. There's additional testing that's done there to ensure that the animals are maintained at SPF status, specific pathogen free. There's baseline data collection done, setup of ground controls, the baseline dissections are performed close to launch. I'll talk about that a little bit more. And then the flight animals are selected, loaded, and turned over for launch. They're transported to the space station on SpaceX Dragon, in this case SpaceX 17, mission 17. Ultimately, there will also be flights of rodent missions on Northrop Grumman Cygnus missions as well. They are carried up in what are called transporters. I'll show that here in a moment, with environmental conditions maintained in the vehicle to support these animals in a high quality manner. It takes roughly 2 to 3 days, just depends on sort of orbital mechanics, to rendezvous and berth with the space station. And then they're transferred to the space station within the first 24 hours after their birth. Once on space station, the husbandry is performed. They are actually transferred to new hardware called habitats. Again, I'll show you in a second. They are given new food every 7 days, water replenishment as needed. And they're also checked for health daily. Most of the time that's done with video observation. Sometimes the crew will do that. There are obviously opportunities during the transfer of the animals from the transporters to the habitats to get direct video and close-up views of each individual animal. But then in addition, on space station, there are a number of operations that can be performed. Again, this is generic, intended to be generic, sort of general concept of operations. Not all operations will be done on this particular experiment, but the Life Sciences Glove Box is now up on space station and available as a good quality facility where manipulation of animals can be done as needed. There are video observations done from space station that can be used for behavioral assessments. There are, in fact, various measurements that can be made. I'll show that in a moment. There are also opportunities for these types of missions to do treatments. We're not doing any on this particular experiment. And then most importantly for this one, I would say, are the dissections that are capable. The crew will be able to euthanize and process and then ultimately collect a number of tissues that we're intending to do from one subset of the animals that are flown to the space station. Then for the return of samples and animals, a number of the animals in this case will be returned live on that same SpaceX 17 Dragon mission. They will be processed in the area vicinity of Long Beach, where they actually return and then are transported. In this case, we're going to be working with the Scripps Research Institute in San Diego to process the animals. And in addition, tissues and carcasses and blood from the animals that are euthanized on board the space station are also returned, because they are processed after the return of SpaceX 17. That means they'll return on SpaceX 18. They'll return on SpaceX 19. For post-flight processing, the live return mice, as I mentioned, will be processed at Scripps Research Institute in La Jolla in San Diego, where there's additional opportunities for testing before dissections. There's also opportunities for some subset of animals to be recovered and the recovery process evaluated, and tissues and carcasses and blood that are returned from space station and ultimately from the Scripps processing will be returned to Boulder and then ultimately to BioServe here in the University of Colorado Boulder, and then ultimately distributed to researchers that are awarded these tissues. Next slide, please. So as I mentioned, the animals, and as I'm sure you can imagine, have to go into special housing. The standard caging that in vivo researchers might utilize in their labs or in their facilities can't, have a lot of, shall we say, gravity dependencies. So there is specialized hardware that's been developed by NASA to support rodents, mice, perhaps rats at some point, but mice for this study in a high quality manner. The habitat in particular is where the animals will spend the majority of their time. On space station, they are group housed, 10 animals per habitat, 5 per side. There is monitoring of temperature and humidity. And in fact, from space station data, we can also get gas composition, so CO2 and so forth. Water and NASA food bars replenishment is possible with this hardware. I'll show you in a moment. There are video cameras that can be used for observing the animals at, frankly, any time, day or night, because there are LED as well as infrared illumination systems. The system provides for a standard light dark cycle. In this case, we'll use a 12-hour on, 12-hour off cycle. And what will be unique about this mission is we're actually also going to be incorporating radiation dosimetry with simple passive dosimeters attached to the hardware so that we can get a sense of the additional, or get a measurement essentially of the radiation exposure that these animals will experience while on the space station compared with ground controls. Next slide, please. Just to give you a better sense, since it's hard to see inside the photo of the hardware, this is, on the left shows a breakaway of that habitat volume. It shows you the locations of the cameras looking into the cage so that you can see the various areas in which animals can live and move. It also shows the location of the food. The food is actually a special nutrient food bar that's formulated and has been used for many years now by NASA for these rodent missions. It is a nutritious food that has been used on many missions, and the mice do very well on it. There are 2 Lixits redundant, so that if a water system fails from one of them, the other one will allow for water. So they have basically ad lib access to food and water. In addition, the Animal Care and Use Committee with NASA requires some form, and it's good practice, some form of enrichment, and this is in the form of the enrichment hut. Essentially is what we refer to it as the blue wire mesh that you see there in the bottom. This allows the animals to have a more confined space that they can get into, they can huddle, they can sleep, etc. And so that's now included and will be incorporated in this mission as well, as it has been on recent missions. In addition, you can see that the transporter in the photo in the upper right picture, that's what's used for transporting the animals up and down on Dragon. Essentially looks very, very similar to the habitat, and in fact it is. The cage environment is really very similar. The big difference is that there's no hut involved in the transporter. The cage is slightly oriented differently, but otherwise the principle and the use of food bars and ad lib water is the same. Next slide, please. In addition to the habitats, of course, the housing is very important for this study and for any rodent study, but there are other facilities and equipment that we use on board space station that we have access to, that the crew has access to. Probably the most important is, or one of the most important is cold stowage. This allows us to process, to euthanize, and then to dissect and collect individual tissues, and then preserve those tissues in a high quality way, maintained at below -80 degrees Celsius, a typical -80 freezer, and maintain that all the way through return and then ultimately shipment out to investigators that are going to be analyzing those tissues. The Life Sciences glove box, as I mentioned, the photo here in the upper right is actually the materials or microgravity sciences glove box. The LSG as opposed to the MSG is a little bit larger. And the nice thing about the LSG is it allows 2 crew members to operate inside the volume at the same time. which will allow us to do more detailed and more precise dissections and a little bit more rapidly by using 2 crew members, as opposed to previously we could only use 1. There are other systems on board that in this particular case we're not going to be using. Bone densitometry, grip strength, mass measurement. As you can imagine, you can't just put these animals on a simple scale. There's a special device for that. But all of these things have sort of pluses and minuses. And for this particular mission, we are not intending to use these measurements for this. We are going to, however, emphasize the dissection process for the animals that are maintained on space station and euthanized there. And so an important part of that will be kits, for example, that are used to anesthetize, to collect blood, to preserve various tissues, vials, etc., and of course the dissection kits that are important. Next slide, please. Okay, so specifics for this mission, as Liz mentioned, the launch date is currently on the schedule for July 8th, of course subject to change, with tentative splashdown and return on August 13th. And for the samples that are obtained on space station, the return would be on CRS-19, which is currently scheduled. That return is currently scheduled in very early part of 2020. Next slide, please. This slide is intended to give you a much stronger sense of the experiment design. So first and foremost, we'll be flying female C57 black mice that are provided by the co-investigator and sponsor Taconic. Half of these will be younger, 10 to 16 weeks of age at launch. Our goal will actually be 10 weeks of age at launch, and half will be older, 27 to 28 weeks at launch. The groups are going to be age and weight matched at the time of launch. And so we go through and very carefully make sure that animals are socially getting along well in their cage with their cage mates, that there's no overall health issues, and that the animals are very healthy. This is obviously both for the flight and ground controls. We split groups. The groups that will be flown will be split between the live return and the ISS terminal. As you can see in the chart to the right, there's 160 mice that will be utilized in the study altogether. 40 will be flown. 40 will be baseline controls. These are animals that are euthanized at the beginning of a launch to establish the basal conditions or phenotypes of the animals prior to launch. And then there's going to be 2 different types of ground controls. The habitat ground control, which will be housed, as Liz mentioned earlier, in the identical same hardware. Essentially the transporters and the habitats, and subject to the same conditions, environmental conditions, as the flight animals. But we will also have a vivarium ground control, which we did in fact set up for the first rodent research reference mission, the first one as well. And the reason for that is that we believe that it's important for some to be able to look at and understand cage effects. In other words, what are the effects of the housing environment itself, and how to interpret any differences that are associated with the housing environment to better interpret what the data that comes from the flight animals. So we have these 2 different types of controls. The intent is to have 30 actual live returned animals from this mission, and 10 that are kept on space station for the euthanization and processing by the crew. Of the 30 return animals, the intent is for a number of those animals to be processed immediately upon return, as quickly as possible, to capture essentially the primary effects of the exposure to spaceflight. But some of those animals will also be allowed to recover, and the idea is to allow them to recover until they are the same age as the terminal groups on space station, and therefore processed at the same time. And that will allow for a direct comparison between animals that were maintained on space station for roughly 60 days versus animals that were on space station for roughly 30 days, but then allowed to recover for an additional 30 days before they're processed. Next slide, please. This slide is intended to show you the types of tissues that we collect. As you can see, it's fairly comprehensive. And even within here, we will sometimes, for example, subdivide tissues. For example, with liver, we would collect multiple lobes. With blood, we would subdivide serum to maximize the utilization of that serum. So there are multiple samples that can be collected. And this is not even intended to be comprehensive. In fact, for the first reference mission, we collected more than is shown here. And we would obviously try to utilize the solicitation and the proposals that are awarded to define additional tissues as required or as desired by various investigators. So this is not meant to be a comprehensive list. It's meant to be to give you a sense of the types of things that we collect. In addition, what's shown in blue here are for the ISS terminal group, the ones that will be euthanized and processed on orbit, the specific tissues that will be collected and individually preserved. So for example, for the left, the muscles and bones, those are going to be actually collected left on the hind limbs. We're taking the entire hind limb from the animal, and then in the case of one side, we're going to preserve that in RNA later for better gene expression, and the other side will be preserved in 4% paraformaldehyde, so fixed essentially, and preserved for morphological analysis. Both eyes will be preserved in the same manner, one side in PFA, the other side in RNA later. The brain, the intent right now is to hemisect the brain and preserve one side in PFA and one side in RNA later, and so forth. Same with the kidneys. Obviously, the individual tissues, spleen, liver, and heart would be preserved in RNA later. In this case, is our intent. The blood is collected through cardiac puncture, is separated on space station, centrifuged, and then frozen in its tube so that the blood pellet and serum are separated at that point, and then the serum can be further aliquoted as necessary on the ground after return. Next slide, please. In addition to collecting tissues, clearly there's an opportunity here to collect additional data that would be and could be utilized by investigators to help interpret observations they see with individual organ systems or tissues or samples, but also to get a sense of things like animal behavior. So one of the things that we're trying to accomplish with this, as I said early on, is to validate spaceflight models and how they can be utilized and how they can be understood and interpreted. So behavioral assessments are an opportunity here for us to help understand how these animals respond to this environment and how they recover from this environment, both in regard to things like anxiety-like behaviors, motivation, cognitive functioning, as well as the physiological behaviors like grip strength or sensor-motor behaviors and so forth. Clearly, obtaining animal weights and organ weights and so forth will be done for the animal weights, obviously pre-flight and post-flight, so that those comparisons can be made. For organ weights, we would collect those at time of dissection, and again, make those available to people. And body composition as well. So for this first reference mission, we used an MRI system that can give you body composition data as well. All of this is made possible through our partnership with Scripps Research Institute. They have a behavioral core center there that has provided a lot of this testing for us with the first reference mission, and we expect to use that for the next one as well. Next slide, please. The specific timeline for this mission, again, launch on July 8th, takes a couple of days before it arrives. The animals will be transferred. Approximately, this is a little bit longer duration birthing opportunity than is typical with SpaceX missions. It will be approximately 34 days. At that point, the animals will be transferred to a transporter and returned on the same mission. The animals that are kept on board will be processed at about 60 days into the mission. And obviously there will be identical ground controls as well as the vivarium controls that will be processed at the same time as the animals on orbit. Next slide, please. I'm not going to go through this. This is really just for reference purposes. It allows you to look at and perhaps even ask questions about previous missions that have flown. These are the missions that have been flown to date on the US side. There are additional missions that have been flown by the Japanese space agency, but these are NASA supported missions, many of which, as you can see here, are ISS National Lab sponsored missions. And some of the details of specific strains and animals. And as you can see, Taconic has been a major provider of the animals for almost all, or actually for all of the ISS National Lab sponsored investigations. Next slide, please. And finally, I just throw this up here. There are a number of publications and presentations that you can access to gain additional information and insight into some of these past missions. Some of them are not yet published in peer-reviewed journals, but I do happen to know that for RR1 and 2 and RR3, that publications are being in various stages of preparation and should be out within, I would say, a few months. RR5, I think, as well. And so these missions are beginning to find their way into publications, but in the meantime, you can certainly access presentations that were given at ISS R&D. There are also presentations that were given at last year's American Society of Gravitational and Space Research that I'm aware of. So there is data that you can access to help you in preparing your specific proposals. And with that, I will turn it back to you, Liz.
Thank you, Dr. Stodiak. Next, we'll hear from Dr. Gretchen Cusick, our co-investigator in this endeavor from Taconic Biosciences, where she serves as Associate Director of Scientific Services. Gretchen, and next slide.
Thank you, Liz. Taconic's mission is to improve quality of life by providing the right animal model solutions to drive critical research from project to cure. Taconic delivers this superior value by making its customers the focus of everything the company does. For those of you that may have not heard of Taconic Biosciences, we are a global company providing over 65 years of rodent research model products, services, and solutions supporting scientific advancement and paving the path to cures. Next slide, please. We provide a full spectrum of solutions for every stage of drug discovery and basic research. We work with all types of customers, including large pharmaceutical companies, biotechs, academia, and the government. Taconic offers off-the-shelf models for both mice and rats, which include traditional inbred strains like the Black 6 mouse model, commonly known as B6, other inbred strains such as BALC, 129, FBB, and outbred mouse strains like the Swiss Webster. Taconic provides access to an immense variety of genetically modified models, some of which are referred to as humanized models, as they are modified to express human versions of a specific protein or antibody that replaces the mouse version. This is important in many preclinical studies. Taconic also offers customized services, which includes generating genetically modified rodent models specific for a client's needs. Perhaps there is a new target or gene of interest, but there is not a model available, so Taconic can generate one for you. Of course, once you have a model, it needs to be scaled up in breeding to generate the cohorts needed for your research project. Taconic has a project management group that oversees custom breeding project execution, working in close coordination with the investigator to meet project goals, timelines, and budget. We offer embryology services, including rederivation to reset model health status, cryopreservation to secure your model, along with many value-added services, including but not limited to genotyping, health testing, surgery, and tissue collection. Next slide, please. Taconic has a history of supporting rodents for spaceflight that goes back into the 80s, where Sprague Dawley rats were flown to validate new equipment that NASA developed to hold rodents on the shuttle for extended periods of time. The quality of animals, specifically the high health status of the rodents maintained at Taconic, was the initial determining factor in choosing Taconic animals for spaceflight. Many flights followed with other Taconic models utilized. Next slide, please. More recently, from 2014 onward, Taconic has supported 6 missions sending mice to the International Space Station. 5 of these studies were hypothesis-driven, led by either pharmaceutical or academic investigators, as Dr. Stodiak has outlined in his previous slide. Taconic's main role here was our project management team that worked very closely with the investigators, along with BioServe and the ISS National Lab scientific team, to generate and prepare the mouse cohorts for spaceflight. This varied across the missions, as some missions utilized Black 6 mice, and others utilized the Balb C strain. The only mission to utilize a custom genetically modified model was the Rodent Research 1 mission, which required even tighter coordination and project management to produce a large group of animals with the requested genotypes of mutant and wild-type littermate controls. These were to be within a single week of birth, while also ensuring they were the correct age at the time of launch. This brings me to my next slide regarding project planning. Next slide, please. There is a lot of upfront planning and many parties involved when prepping a cohort of animals for spaceflight. This slide outlines some of the key considerations, starting with animal model selection. Will the rodent model be a commercially available inbred strain like Black 6, or a genetically engineered model that may be commercially available, bred at your facility, or generated at a third party? In general, sourcing a commercially available inbred or outbred strain lends to ease of overall project planning, as access and availability is generally easier with these traditional models, as large cohorts of traditional models can be purchased off the shelf. Genetically modified models may require scale-up and breeding to produce the large cohorts within a tight age range, or even rederivation followed by scale-up rapid expansion, which will take more time up front. So you'll need to plan at least a year prior to the launch date to ensure your cohorts are ready. This planning also involves setting your cohort goals. How many animals? What genotype? What sex do you need? Do you need wild-type controls? What is the target age at launch? Will aging of the animals be required? You'll also need to consider planning for a backup cohort to be available in the event of launch slips or scrubs to ensure there is a cohort within age spec at the time of launch. This is easier when working with readily available commercial models, but will require extra planning if working with a genetically modified custom colony. Health status is another important consideration, as there is a minimum health standard acceptable by NASA for spaceflight. This may require specialized health testing, rederivation, or expansion of your custom lines. For commercial inventory, Taconic provides animals off the shelf that meet NASA health requirements, while rederivation and rapid expansion options are available if an investigator's model does require a health status reset. There are additional services for considerations such as animal identification. Previous missions utilized RFID microchip implants, and sometimes tail tattoos were administered to the cohorts for animal identification. Additionally, ongoing body weight monitoring is carried out to ensure the selected animals are within a tight age range, that selected animals within the tight age range are also within a tight weight range for the least variability. Next slide, please. This slide is an overview of the health profiles available at Taconic, and specifically focusing in on the ES, or the excluded flora health status. This is the health status in alignment with the NASA exclusion list. This profile excludes all viruses and parasites, and for bacteria, it is the opportunistic pathogens that are the differentiator compared to other health profiles, such as a specific pathogen-free or a murine pathogen-free health profile shown here. Next slide, please. So Dr. Stodiak presented a very detailed slide that outlined the cohort specifications for Rodent Research Reference Mission 2, and this slide is here to review the cohort specifications. This mission will utilize Black 6 mice of the C57BL6N-TAC substrain from Taconic's colony at excluded flora health status to meet the NASA requirements. Links are provided here for information on this model, which includes a white paper titled The Most Important Mouse in the World, and that discusses the importance of the Black 6 model in preclinical research, owing to its utility in metabolic studies and its popularity as a genetic engineering platform. For the cohort, there will be 2 groups of Black 6 females, an old or aged cohort, and a young cohort. The old and young groups will be separated into flight, ground control, and baseline groups for these studies. For the rodent research reference mission, for this one, the old group is currently housed now at Taconic for Aging for the end of December 2018, and the young group will be sourced from a single week of birth at the end of April 2019. These mice will be a minimum of 26 and 10 weeks of age per group, respectively, by the July 8th, 2019 targeted launch. Back to you, Liz.
And thank you, Dr. Cusick. Next slide. This schedule is what we anticipate following for the mission. Answers that you submit today, either via email or in the next few weeks via email, will be posted to our solicitation website by about mid-April. Proposals are due by 5 p.m. Eastern, May 17. We expect to have reviews and selection complete by June 10, and we anticipate announcing awards during the week of June 17. Again, that is for a July 8 launch target. One additional form will be added to the RFP that is not on our website yet, so I just want to give you a heads up. That form will allow you to include specific information about the tissues and preservation methods that you require for the biospecimens that you are requesting. So that is to be uploaded very soon. Next slide. Of note, we can only award U.S. persons as described in Section 1.4 of the solicitation guidelines. Next slide. I've mentioned the solicitation website a number of times. Again, it's here for you to reference at the webinar. There's one additional informational document, which is a PDF released by NASA that describes, it's essentially a researcher's guide to ISS rodent research, which includes a little bit more information on the habitats and the facilities that are available for rodent research, this mission as well as others. And on the solicitation webpage, there's also a document that further describes the standard housing that is provided on the International Space Station. Next slide. Finally, we do have a few minutes for Dr. Bill McClam, Operations Project Manager at the ISS US National Laboratory, to address any questions that we've been seeing in the chat panel that an answer would behoove everyone to hear at this time. Bill, over to you.
Thanks, Liz. Good morning, everyone. Appreciate everyone's attendance today. It looks like we got about 30 attendees. We have about a half a dozen questions that came in through our online portal, and I'll go through those quickly. And if I didn't fully answer your question, please speak up, and we can address it in the time we have left. And I see we have a couple questions coming in now, so we'll just when I get to those, we'll rather than type answer, we'll talk to them directly. The first question, and you should have all received the question and answer as part of the question feed box. So, but we'll go through them anyway here. First question was how long on ISS before they are returned? The animals are returned. Approximately 30 days, and that was presented on slide 17. And can we have a copy of the presentation? Yes, of course. We'll be recording this presentation and providing a link to this for all participants and other interested parties that could not have made the event today. Then a question, could health impacts on the animals be measured in the experimental design? No, specifically related to effects due to space radiation and solar flares. And yes, these impacts can be measured and will be measured with the use of radiation dosimeters, which are part of this experiment design, and we intend to include in future reference missions as well. Next question, will calvaria be dissected or broken into smaller components? For the first reference mission, the dissections for baseline control and the other ground control animals, and for the live return mice, calvaria were dissected and divided into left and right halves. They're still in work as to whether we can dissect the calvaria as part of the on-orbit procedures. For the terminal dissections, that's currently in work. So if it can't be dissected on orbit, then they would be would be frozen with the carcass and then would be dissected out for post-flight during post-flight processing on the ground. And is someone addressing the application? I think we did address that briefly as far as the schedule and links to the solicitation. If you have any other questions, if time permits, you can certainly ask more. I've got 4 more questions that I don't have written answers to, so I'm going to go through these. And if I don't have the answer, Liz or Louis or Gretchen can chime in. Are we eligible if the organization is located in the U.S. or working remotely from non-U.S. location? I'd like to confirm that with our compliance folks just to be sure of the exact answer. So stand by on that. What funding is available?
I'll chime in. If you could email, Bill, on that one, the answer or the questioner should submit the same question to info@issnationallab.org. That's one word, issnationallab.org, and we'll answer that individually.
Thanks, Liz. The next question, what funding is available to support assessments of the in-flight versus post-flight specimens? This solicitation was specifically designed what is provided are the tissues and the animals for analysis. So if your project is selected, that is essentially the award. There is no monetary award for this solicitation. Next question. Our group has 3 NIH-funded investigators. We have been discouraged by the length and complexity of the application. Any thought of streamlining proposals? We will certainly take that into consideration. I think for this particular solicitation, we don't have the ability to do that, particularly with the short turnaround time. There were some reasons for this one being a shorter turnaround time than desired, but for this particular proposal, we won't be able to streamline the proposal process. Next question, could specific diet inclusions be part of the project as a requirement? Let me scroll down. As a requirement for some studies. For this particular solicitation, no, we have a standard feeding protocol that's part of the approved protocol, so we can't change that part of the experiment design. And the last question, is it possible for the same tissue to be separated for projects in multiple labs, or is it a one tissue, one lab type project? So the form that Liz had referred to about identifying the specific PI requirements will identify. We hope that you can identify the minimum number of samples you would need, so we can distribute them. For instance, if there was an investigator that wanted liver tissue, but not necessarily from 20 animals, then there's an opportunity to distribute to multiple labs, and that is specifically why we're asking these questions up front with this additional form, so we can best manage the selection and distribution of tissues, because we want to maximize utilization of these tissues across the community.
And Bill, to chime in, this is Louis. I think they're also asking in regard to, can they request multiple different organs or tissues, and the answer to that is certainly yes, if that is what's needed to meet the research objectives of your particular proposed study, or if there's multiple studies from an investigation team, I think that's probably acceptable as well.
Okay, thanks for that clarification, Louis. So it looks like we do have about 10 minutes left in our time. So if there are specific questions from anyone of the attendees today that if they would like to ask the question, please feel free to you can either type in your question, or I believe you can raise your hand to ask the question, and we can unmute your mic. Well, while we're waiting, I'll just ask if Louis or Liz or Gretchen have any additional thoughts to add before we close out today, or after we get the last questions. I think we have one.
I have
one hand raised from Dan Blaitler.
Nope.
Nope.
No, I'm sorry.
Okay.
I don't. By accident. My apologies.
Okay, no, it's okay. Well, I'm glad we could answer everyone's questions within the time allotted, but this certainly doesn't end the question and answer session. You can certainly ask us more questions through the links on the last page that Liz provided. Actually, I think we've got a couple more questions that just came in. Let's see. What percentage of submitted proposals are approved typically? Would you email a link to the application? Yes, of course, we'll send a link to the application. But as far as percentage of submitted proposals, since this is only our second reference mission, it's hard to quantify that, and it's really going to be dependent on the specifics of each of the proposals received and how we can best distribute. We would certainly like to select as many as possible, because again, we're trying to maximize utilization of this very rare opportunity.
Hey, Bill, it's Gretchen. Can I say something?
Yes, please.
So this is, I guess, for Bill, Liz, and Louis. There was that one question about a special diet, and obviously this is a rodent research reference mission. Everything's already set, and I don't even know, if something like that would be possible in a custom study similar to what has been done in like the rodent research 1, 2, 3, 6, those missions that Taconic sponsored, where it was more investigator-driven, custom hypothesis-driven studies. If someone's interested in doing something like that separate from this mission, is that still possible, and who do they reach out to learn more?
Bill, I'll take that one. This is Liz Warren again. We hope that this webinar has intrigued you and been something that encourages you to either apply for these specific rodent tissues, or if in the meantime you are thinking of a more customized rodent research mission, for example, if you want to have a customized diet, there is a possibility you can submit an unsolicited proposal to the ISS National Lab at the websites shown on the screen now. And there is certainly a opportunities for custom rodent research missions. So we do invite you to think about future opportunities. And again, we have a rolling submission where you can submit an unsolicited proposal at any time. So we do invite you to also consider that as an option for obtaining spaceflight tissues. And with that, I think
And
Bill,
Liz, I have one more follow-up.
Yep.
Yeah, I have one more follow-up talking to our folks here regarding the number of proposals and awards for the last solicitation. We received we had 12 proposals that were responsive to the solicitation, and of those 12, we did award opportunities to 11. So again, that's all going to depend on how we can best distribute the tissues to meet everyone's needs. If we have 10 PIs that all want the same bone tissue, that may be hard to do, but there are a lot of different opportunities, tissues within each laboratory that may still allow us to award multiple awardees. So I encourage you to...
And Bill, this is Lewis again. I think it's also worth pointing out that there are, in fact, tissues available through the first reference mission that are still available and are being archived, preserved, etc., as well as potentially data that can be obtained from these earlier missions if you want to pursue that, either in preparation for this solicitation or separately, down the road.
Thank you, Lewis. That's a very important addition to the attendees here.
And with that, I would like to thank everyone for their time today and encourage you to consider sending in a proposal. We look forward to interacting with you more if you've got additional questions. And again, thank you very much for your time.